Hi everyone, it's Mr. Sinti, and today I'd like to talk to you about photosynthesis, but in particular, I'd like to talk about the light reaction. Yeah, the light reaction obviously involves the sun, but what's fascinating about it is that it's one of the most interesting and important biochemical processes in all of life. And so there you go, taking it up. So the light reaction is one of two parts of photosynthesis. You can call the other part the dark reaction. I like to call it the Calvin cycle after Melvin Calvin, Nobel Prize winning botanist, biochemist from the University of California. It's the pathway in which sugar is produced in photosynthesis. It's called the dark reaction, but in fact, it's reliant upon the light reaction's products. So it's a little bit of a misnomer to call it the dark reaction in general because it only occurs during the light of day. And so this particular video is going to be on the light reaction. And so what it's going to do is it's going to take in light in the form of the sun. And so as I mentioned, photosynthesis is divided into two processes. And so we have the light reaction, which involves the sun, and solar energy is converted to chemical energy. And it's not the chemical energy that you might be thinking of, glucose. In fact, it's the chemical energy of ATP and a reduced coenzyme called nicotinamide adenine dinucleotide phosphate reduced. How's that for a mouthful? We'll get into it. And so the Calvin cycle is the second part, or the second process. It's where the plant in the chloroplast is able to incorporate CO2 in the stroma and convert that into sugar. And in order for the Calvin cycle to work, it needs NADPH and ATP to do that, and that comes from the light reaction. So the dark reaction requires the light. And so I like this diagram. It's kind of a summary. It shows here here's a plant cell, and these green bodies, or these plastids, are called chloroplasts. And in particular, there's a close-up outer membrane, inner membrane, stacks of grana. Each one is called a thylakoid. And so the light reaction, which is what this video is about, is going to occur in the, it's going to occur in the membrane of the thylakoid. So this is where the light reaction's occurring, in the thylakoid membrane. And so the inputs or reactants for the light reaction are, of course, light. Then we have water. Water is used in the light reaction. Water is oxidized to produce oxygen. As it turns out, we'll look at the detail of that. And also you need NADP+. It's sort of the cousin of NADP. NAD+, it just has a phosphate on it. And of course you need ADP as well. And so the products of the light reaction are principally ATP and NADPH. And then those energy molecules will be used in the stroma, which is the fluid part of the chloroplast, to incorporate CO2 to produce sugars. All right, so there you have it. It's a two-part process. So the light reaction is going to convert solar energy to the chemical energy of ATP and NADPH. And that's happening right in these little structures here called thylakoids. And so they're really thin right there. So they're stacked up like that. And so the sunlight is going to get picked up by the chlorophyll in the thylakoid membrane. So it's all about the thylakoid. This video is all about the thylakoid. You're like, all right, what is a thylakoid? It's kind of a simple structure. It's a phospholipid bilayer that forms a little pouch. So this is, again, a three-dimensional thing. And what happens is it's very receptive to picking up the energy of the sun. And that is because in the phospholipid bilayer, there's pigments. And those pigments can absorb sunlight. And so light travels in rhythmic waves. It's picked up by the leaves or any green parts of the plant. And if you weren't familiar with this, this is the electromagnetic spectrum. And what we're talking about in photosynthesis is this narrow region of the electromagnetic spectrum known as visible light. So it ranges from high frequency violet to low frequency red. And so this higher energy area right in here, inversely proportional, is a preferred frequency for plants to photosynthesize. And so this is what I was talking about a little bit earlier in terms of the thylakoids. The thylakoids themselves have a phospholipid bilayer. And as you can see, let me get silly with this and say, let me draw a thylakoid like that. And so you can see that with the green line is actually a phospholipid bilayer. And then again, embedded in there are these these chlorophyll pigments. They're kind of like musical notes, like that. And what's interesting is they have this perforin ring. And this perforin ring has magnesium in the center. And that is what's going to actually be capturing the sun. And so the perforin ring can be different in chlorophyll A versus chlorophyll B. So that's something to point out. Okay, so here's a three-dimensional model of chlorophyll. And let's take a little journey over here for a second. I have something to show you with regard to this. Like, take a look at this. So chlorophyll is a really cool looking molecule. Let's see that again. It really looks like a lollipop right here. And here's the perforin ring, and then there's that magnesium in the center, which is held together by those nitrogens and this carbon. And so it's really conducive to capturing sunlight. And so I don't know, it's kind of neat that way, that the structure of it is designed to do what its function is, which is absorb sunlight. And so that's kind of neat. So what's interesting, in the thylakoid membrane, you have a cluster of several hundred chlorophyll molecules. And that's what this big green thing is right here. It's a big photosystem. And as it turns out, there's two photosystems in photosynthesis. Apologize for this, but the very first photosystem is referred to as let me write that there. Whoops. Sorry. The first photosystem in photosynthesis is Photosystem II. And that's that's always a bugger, but I'm not sure the background specifically, but it might have been, I don't know, it might have been the fact that it was discovered second, although it goes first in photosynthesis. So this is known as Photosystem II. And there's a bunch of chlorophyll, so if I were to draw them in, like that, and in the center of this thing is the reaction center, which is often chlorophyll A. It's going to be the one that it's going to get right here. Let's make this one chlorophyll A. There's chlorophyll A, and it's going to be the one that's going to get its electrons excited. And when the electrons are excited, what's going to happen is adjacent to chlorophyll A, or adjacent to photosystem II, is a bunch of proteins embedded also in the thylakoid membrane that have electron wanting coenzymes. And so they're electronegative molecules that are embedded in the membrane as well, and they're going to steal away the electrons, and so they're going to begin an electron transport chain. So there's a primary electron acceptor, which is shown here in this purple, which is going to remove the excited electrons. So again, let me illustrate that. So here's the chlorophyll A, and when the electrons get stolen away, okay, remember, two hydrogens contain two protons and two electrons. So this coenzyme is going to get reduced, and in so doing, it's going to cause the oxidation of chlorophyll A. And so this is known as the photooxidation of chlorophyll A, because light was the one that actually caused the excited electron to move over in the first place. So photooxidation of chlorophyll A starts the light reaction. All right, and so that's pretty cool. So what's happening is photosystem II, again, kind of funny because it starts first. Photosystem II absorbs the light. The excited electron is captured by this primary, which means first, electron acceptor, but there'll be others. So what happens is it leaves the reaction center, and so when the electrons leave, they're considered to be oxidized. And so there's an enzyme then at the base of the photosystem that actually extracts electrons from water. This is rather critical. So water, shown right here, is obviously needed by plants. So if we back it down in terms of complexity for a second, everyone knows that plants need water. And it's like, well, this is one of the main reasons why they need it. They use water, H2O, as a source of electrons. Because if you think about it, if chlorophyll keeps getting its electrons stolen away, right, in here, if they keep losing electrons, ultimately they're going to run out. So water provides, as you can see here, provides those much needed electrons. And in so doing, if it loses electrons, water is oxidized itself, loses electrons, and who's being reduced? Chlorophyll A. And in so doing, it becomes atmospheric oxygen ultimately. And this kind of leaves the chloroplast. It goes into the spongy mesophyll of a leaf and then out the stomata and into the atmosphere. And so we breathe it in. This is where the oxygen that we're breathing in right now while we're watching this video is coming from photosynthesis, and water's giving it up. And so that's pretty awesome. It's kind of a waste gas if you think about it that way, because the most important thing is the electrons. And so oxygen is liberated in the light reaction. And so there's two photosystems. As I referred to it, this very first one in photosynthesis is called Photosystem II. So that's right there. Photosystem, let me do that. Photosystem II is right here. And then this guy over here is Photosystem I. Okay? And so as it turns out, the way we categorize Photosystem II and I is by its reaction center. So the chlorophyll in the reaction center of chlorophyll, Photosystem I, prefers a nanometer of 700. Sorry for leaving hanging on that. So photosystem 1 prefers 700. Sometimes it's known as P700. And then photosystem 2, which is the first one, is called P680 because it prefers that. And as it turns out, it has more to do with the proteins than it does with the chlorophyll molecule itself that the whole photosystem is embedded, as it turns out. So this is photosystem 2, photosystem 1. Okay, and so here's a schematic of the light reaction of photosynthesis. Sometimes it's referred to as the Z scheme. When I first learned this as an undergraduate, it was referred to as the Z scheme, right there, if we can sort of draw it that way. I have to admit, I didn't know what was going on while I was learning this. I was like, Z, I don't see it, but I guess it's here. So what's happening is light's coming down, it's striking the thylakoid membrane. And so this is a bit of an abstraction, but I like it. So the light comes down in a photon, bam, 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 photosystem 2. It hits the P680 reaction center of chlorophyll A. It excites the electrons. You can see here it's like shooting up like a firework. And as it turns out, those electrons are then stolen away from chlorophyll. And they're stolen away by oxidative coenzymes, in other words, electronegative. They're oxidizing agents. They steal the electron away. And in so doing, there's a loss of electrons. And so this gets reduced, and then this loses electrons, and then the next one. So it's a cytochrome complex, if you're familiar with that, where each of these undergoes a redox reaction. And so a loss of energy then allows the chloroplast to, and in particular the thylakoid, to scoot protons into the thylakoid lumen. And so they tend to accumulate. And then the cell's able to use that proton motive force to produce ATP. I'll get back to that in a second. So what's happening is another photon, so two photons of light, another photon strikes photosystem 1. It excites the electrons, and they go up into a higher energy state. And so what replaces the lost electrons here is photosystem II. So ultimately, water is providing the electrons. As you can see here, it is producing oxygen. Water is bringing the electrons in. They go up, they come down, and then they replace sort of the hole left behind by the loss of electrons here. And then there's more electron transport chain, and then ultimately they go into the very last protein, which is a reductase enzyme, which is then going to allow NADP+, which is nicotinamide adenine dinucleotide phosphate, to become reduced. And so the energy is then stored in NADH, meaning because it now has electrons that it didn't have before, and then ATP is produced. And also oxygen, of course. Those are the products of the light reaction. And so, again, I'm not much for a cartoon, but sometimes they're a little goofy, to be honest. But this one, I think this one's pretty good. See what you think. So here's this, I don't know, this construction site, and this worker down here is has this big hammer, which is a photon, which has energy, and so he whacks his paddle right here, and it sort of propels in a teeter-totter kind of way. It excites the electrons higher up. And so here is this worker who's then using the electrons and putting them down this mill, spinning the wheel, which allows the thylakoid to make ATP. And then this guy is another photon. He whacks it, and so the electrons then go up here, even higher still, and then they're stored. I like that metaphor, in a bucket, which is being symbolized by NADPH. So kind of cool. So this is photosystem II, photosystem I. You're like, where are these electrons coming from? Where do you think they're coming from? They're coming from water. And so the water is what's providing the supply of electrons, and in so doing, it becomes oxygen. So this is really profoundly important. If you're following this, and I hope you are, water is the ultimate source of energy. That's where those electrons are coming. And light is, I didn't say merely, but it is a catalyst for the oxidative photooxidation of chlorophyll A. So there it is. Pretty awesome. Okay, so this is important too, because this is a look at one thylakoid. So here it is. So it's like a cell within the chloroplast. And so here are these big proteins that are embedded here, and here's the photosystem, right in here, photosystem II, here's photosystem I. So this is what we've been talking about, but it's more literal this way. So the light comes in and excites the electrons, the electron transport chain carries on, and in so doing, the cell is able to use, the chloroplast is able to push these protons in here. And so these protons accumulate in the thylakoid lumen. So it becomes rather acidic and highly concentrated in protons. And then out here in the stroma, it's negative. And so how do you like that? So this dark area is positive and the stroma is negative. That's an electrochemical gradient. And when you create an electrochemical gradient like that, the protons are itching to get out because they're attracted to the negative charge in the stroma, and so they're trying to get out. And so the only way they can get out is through a proton transport. And when they flow down that proton transport and something called the proton motive force, chemiosmosis allows the protons to move through, which then causes this whole protein to spin around and rotate. And guess what? There's an enzyme called ATP synthase, which then is capable of attaching inorganic phosphate to adenosine diphosphate to make copious amounts of ATP. It's just like in the mitochondria. And so, if you're familiar with that, and so it uses chemiosmosis to produce ATP. And so the ATP and then ultimately the reduced power of NADPH is going to be used in the Calvin cycle in the stroma to produce glucose. And so pretty awesome. And so let's take a look at this process in terms of like a little animation. I think that would be useful. And so let's take a look here. And so what you have going is this big green guy over here is a photosystem, here's a photosystem, and then here's the electron transport chain. So the photons of light are coming. And then notice how the electrons are passed from photosystem 2 to photosystem 1. Okay, so those are where the electrons are going. And so the power of the electron transport will facilitate the thylakoid's ability to store up protons. And so it becomes really acidic, and then those electrons, where are they going? They're getting picked up by NADP plus to produce NADPH. And so now the thylakoid needs more electrons, so it uses water as a source, and so therefore oxygen is produced. So the whole point of this is to generate an electrochemical gradient, and now these protons are trying to get out of the thylakoid, and so they can't, so then they end up passing through this protein that is associated with ATP synthase. And so therefore the proton motive force, or the chemiosmosis, allows the thylakoid to produce ATP and NADPH, which is then used by the Calvin cycle to produce glucose. That's pretty awesome also. And so I like this particular video because it's really well produced. And so check this out. Same thing happening. Photosystem, let's calm it down for a second. Some bearings. So this is the inside of the thylakoid. Down here, here's the stroma. This is photosystem 2, photosystem 1. This is the electron transport chain here and here. Let's take a look. And so what's happening, if we can close up and photosystem 2, it'd be great to see what's happening. So those little starbursts coming down as light. So let's get a real detailed look of what's happening in photosystem 2. You can see it over here. Water is giving up its electrons. See, water is giving up its electrons, and then the electrons are then being moved over to what's known as the, let me pause it. This is the primary electron acceptor. So chlorophyll A is oxidized, and then this guy is reduced, and in so doing, these protons get moved into the thylakoid lumen. And so water is then, again, providing the ultimate source of electrons. The electrons get moved along the electron transport chain, and as they move along the electron transport chain, it allows the thylakoid to be able to scoot those protons inside. And so look at how many there are. There's so many protons. And then again, when this photosystem loses electrons, they're replaced from photosystem 1. Let me back that up just for a second. And so there they are, another photon of light. So the electrons move along, and then who's that little red car? That's NADP. And so now it's reduced into NADPH. And so now all of this, the point is so that these protons can flow back out of the thylakoid to produce copious amounts of ATP and NADPH. And so that's pretty awesome. Right over here, when they flow back out, it causes this to rotate, which is ATP synthase. And so that's, sure, it's complicated. There's no question about the fact that it's complicated, and but it's also very interesting. And so keep your head up in terms of understanding this. And so here again is a summary of the whole process. The two photosystems work together. The electrons flow in what is known as non-cyclic or non-cyclic. And I say non-cyclic because they flow from water. Oops, they flow from water. Let's do that, which is shown here. I'll just use this picture. So the electrons flow from water, and here they are. And in so doing, it becomes oxygen. The electrons flow from photosystem 2 through the electron transport chain, like this, to photosystem 1. And then the electrons come up here like this, and then ultimately they're taken away in that bucket of NADPH, if I can write that, NADPH. So it's non-cyclic. They actually just leave. And so the point is to produce NADPH and then to produce a copious amount of ATP, as we were talking about. And so this is very similar to the mitochondria, as I was referring to previously. notice in the mitochondria, in the inner membrane space, that's where the protons are. That's where it's really positive. So it's positive there, negative in the matrix. And in the chloroplast, it's positive in the thylakoid space and then negative in the stroma. But the same thing's happening in the mitochondria and chloroplast. Isn't that fascinating? Because sometimes people think that respiration and photosynthesis are completely opposed to one another, when in fact they're employing the same electron transport chain in order to create a proton gradient, which then allows them to produce ATP. So that's pretty awesome. I think it's powerful. So the chloroplasts and the mitochondria both generate ATP by the same mechanism, chemiosmosis. And so, as I was saying, the electron transport chain then pumps those protons across, and therefore, when the protons move past, come back, they produce ATP. And so the mitochondria then transfers that chemical energy from food into ATP, but in the chloroplast, it uses the light to make ATP. And then it's going to use that ATP to produce sugar. Okay, so that's a little bit different. So this is kind of an interesting trivia thing, but the proton gradient's pretty extreme. And so when in the light, when illuminated, the pH in the thylakoid space gets pretty acidic. And so that gives the thylakoid the ability, though, to then produce ATP on the other side of the thylakoid. So that's kind of neat. And so let me contrast this. I was mentioning non-cyclic when I was referring to the flow of electrons between photosystem II and photosystem I, and then ultimately to NADPH. As it turns out, sometimes the concentration of NADPH is really high in the stroma, and there's really no need to make it. But the cell really always could benefit from making ATP. And so, as it turns out, there's an alternative to the non-cyclic electron flow. Although it's the predominant way in which photosynthesis takes place, but there is something called cyclic, and so let's talk about that. And so again, sometimes the NADPH is not needed in the Calvin cycle, but yet there is a need for ATP. And so under certain conditions, and those were the conditions, you can have something called cyclic electron flow. And all of that is that it involves photosystem I. And as it turns out, when the light comes down, let me sort of animate it to make it maybe more friendly. So when the light comes down and it strikes photosystem I, which is kind of funny, and it's the P700, what happens is the electrons get excited, and instead of it being pulled along the electron transport chain and then ultimately reducing NADP to produce NADPH, what will happen is the electron transport chain carries the electrons down, which then allows the protons to accumulate, which then allows the plant to make ATP. But watch this. Instead of it going to NADP reductase, what happens is the electrons flow back to the reaction center. And so it replaces itself. So the electrons go up, and they go beep, 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 down the stairs, and then they come back to chlorophyll. And so it's cyclic. This is why it's called a cycle. So it's ATP is produced in a cyclic photophosphorylation. Photophosphorylation. It's a big word. Photophosphorylation, as opposed to non-cyclic. Okay, non-cyclic was when the electrons flow from water all the way BBB to NADPH. Okay. And so non-cyclic is able to make ATP and NADPH, whereas cyclic just makes ATP. Okay, and so the reason being is that the Calvin cycle needs a little bit more ATP, and oftentimes NADPH is in high enough concentration. Although, let me emphasize this again, non-cyclic photophosphorylation is the more dominant and more often occurring. Okay. And so, again, just to summarize the whole light reaction, the whole point of it is to, and I'm going to circle this for emphasis, the point of the light reaction is to make ATP and NADPH. That's right. And it employs an electron transport system to do that. And as a byproduct, of course, oxygen is produced. Now, why do you need these things? Well, you need them in the Calvin cycle to produce sugar. And so in the light reaction, chlorophyll, and the help of that coenzyme, nicotinamide adenine dinucleotide phosphate, NADPH and ATP are then going to be used in the Calvin cycle. And so I mentioned Calvin cycle many times, but it needs the ATP generated by photophosphorylation in order to make sugar. And so I hope that entices you to watch on further constructing a video on the Calvin cycle. And so I hope you enjoyed this video on the light reaction of photosynthesis. Thanks for watching.